Hi dear students, good morning. Welcome to my class. Today I would like to discuss the history of biotechnology. In the history we can see that thousands of years before peoples were using biotechnological techniques for their purpose, but they didn't name their working field as a biotechnology. The term biotechnology is coined by an Hungarian agricultural engineer called Chronological in 1919 to describe a technology based on converting raw materials into a more useful product. And the history of biotechnology we can classify into three. That is, first one is the ancient biotechnology. That means ancient biotechnology means biotechnology were used before AD 1800, and classical biotechnology means the period of biotechnology between AD 1800 to 1945, and the modern biotechnology means the year 1945 to the present. If you are checking the history, we can see in BC 6000, Sumerians and Babylonians were used yeast to prepare beer, and in Egypt in BC 4000 they were used yeast to prepare bread. In BC 420, the great philosopher Socrates hypothesized on the similar characteristics between parents and their offspring. And in BC 320, Aristotle, the Greek philosopher, theorized that all inheritance originated from the father. In AD 1665, Robert Hooke invented the compound light microscope, and using that microscope he observed the cells in cork. And in 1675, Antony van Leeuwenhoek discovered cells using simple microscope, and he have seen bacteria, protists, and red blood cells. In 1701, Giacomo Pillarini found that the deliberate administration of smallpox could prevent its occurrence in later in life, especially in children. That's why most of the scientists were considered he has to be the first immunologist. We know that Edward Jenner is the father of vaccination. Jenner made an observation that the milkmaids were resistant to immune to smallpox. Jenner postulated that these milkmaids were getting blisters from the cowpox. That's why they are getting immune to smallpox. And he tested this hypothesis by inoculating cowpox blisters into a small boy named James Philip, and he got immunity against smallpox. And this was the first vaccination. That's why that's why Edward Jenner is considered as the father of vaccination. And in 1856, the great scientist Louis Pasteur suggested that microbes are responsible for the fermentation. In 1863, Louis Pasteur discovered the method of pasteurization. In pasteurization, he has heated wine enough to inactivate the microbes present in the wine, and from that, he realized that this procedure will kill the microbes, inactivate the microbes present in the wine, and it did not affect the flavor of wine. If the microbes present in the wine is not inactivated, then this that wine will convert into sour wine. In 1865, Gregor John Mendel, an Austrian monk, suggested the law of heredity. He anticipated that invisible core units of information were responsible for noticeable characteristics in organisms. He called these factors later which were called genes. And here his work was mainly carried out on pea plants. In 1902, Walter Sutton discovered the chromosomes. And Walter Sutton found that chromosomes, that means paired chromosomes, contain certain elements which are transferred from one generation to another. In 1907, Thomas Hunt Morgan he started a work on fruit flies, and he would reveal that chromosomes have a defined role in heredity, and additionally he discovered the mutation theory. In 1909, William Johnson used the word gene, which means the carrier or transporter of heredity. And he also coined the term genotype and phenotype. Genotype means the genetic constitution of an organism, and phenotypes describe the morphological characters of organism, that means the external features of the organism. In 1912, William Lawrence Bragg discovered the application of X-rays in the determination of molecular structure of crystalline substances. In 1928, Alexander Fleming first discovered an antibiotic. Alexander Fleming studied an old culture of bacteria infected with fungal growth and found that it did not show any bacterial growth surrounding a piece of mold in a petri dish. This breakthrough gave birth of antibiotics era. And the first discovered antibiotics was penicillin, and it was produced by Penicillium notatum. During the period of 1945 to 1950, for the first time, the animal cell culture were harvested in laboratories and giving birth to the field of animal tissue culture. In 1947, Barbara McClintock, on her first study on maize, she first demonstrated the transposable elements known as jumping genes, and has a capability to move or jump from one side to another side of a genome. In 1950, Erwin Chergaff discovered that in a double-stranded DNA, number of adenine will be equal to number of thymine and number of guanine will be equal to cytosine. That means the number of purine will be is equal to number of pyrimidine. Later, it was termed as Chergaff's equivalence rule. In 1953, James Watson and Francis Crick described the double helical structure of DNA. and they were published the paper describing the structure of DNA in Nature Journal. And this structure of DNA is known as Watson and Crick model of DNA. In 1957, Francis Crick stated the central dogma of molecular biology, that means how the DNA functions to construct protein. He explained that DNA is undergo for transcription and produce RNA, and its RNA undergo for translation and produce protein. In 1966, Nirenberg, Mattei, Ocho, and Ocho cracked the genetic code. They reported that a genetic sequence of three nucleotide bases, they called as a codon, decide each of 20 amino acids. This was the finding. They cracked the genetic code in 1966. One of the major findings were reported in 1972. The first recombinant DNA molecules you developed in 1972. Paul Berg at Stanford University, who first developed recombinant DNA technology, a method for insertion of genetic material from one organism into another. For this technology, he utilized resistant enzymes to cut the DNA into fragments, and he also used ligase enzyme to join these DNA molecules. In the history of medical biotechnology, 1978 is one of the major year because the first recombinant insulin hormone is developed in 1978. The Genentech company announced that its laboratory had achieved the synthesis of human insulin, humulin, using rDNA technology, and they synthesized this human insulin in bacteria. In 1980, Carey Mullis developed a technique named polymerase chain reaction, and this method and this tools used for multiplying DNA sequences in vitro using the polymerase chain reaction, that's called as PCR. And nowadays this technique is widely used for all sectors of biotechnology like medical, agriculture, environment, industrial, almost all field of biotechnology nowadays this technique were used. In agriculture and food biotechnology, 1987 is one of the important year. In 1987, the first field test of genetically modified food plants are approved and for virus resistant tomatoes. In the history of biotechnology, one of major breakthrough research was started in 1990. Beginning of the Human Genome Project is happened in 1990 with the goal of objective to plot all the genes present in the human body, and the expected cost was around $13 billion. In 1996, the yeast DNA was sequenced. That means the first eukaryotic organisms DNA was sequenced in 1996. Yeast Saccharomyces cerevisiae is the widely used organism in as a model organism in molecular biology, and it's also used in industry for the fermentation, and also is used in food industries. In the history of biotechnology, a mammal was cloned firstly. Dr. Ian Wilmot and his research team created the first true clone sheep Dolly. The researchers at the Roslin Institute in Scotland announced that they had cloned a sheep called Dolly from the cell of an adult female. In the year 2000, the first plant genome was cracked. The plant Arabidopsis thaliana's genome was sequenced in the year 2000. And this plant was widely used in molecular biology, biotechnology, and in botany as an experimental or model organisms. In 2001, one of the major breakthrough research was completed in 2001. The first mammalian genome was sequenced in the year 2001. The full human genome was sequenced in this 2001. In 2004, a pet kitten was cloned firstly, and this cloned kitten was called copycat or CC in short. This was the first pet kitten that was cloned. In the year 2008, the first artificial DNA molecule is synthesized. A Japanese scientist developed the first DNA molecule made nearly entirely of artificial parts. This was the first artificial DNA molecules developed. The first plant-based vaccines were developed in the year 2009. A Canadian-owned Medicago produced the first plant-based influenza vaccine in tobacco leaves. In the 2010, the scientists were synthesized first fully synthetic self-replicating bacterial cell. They named the bacterial cells as Synthia. In May 2010, Craig Institute created the first fully synthetic self-replicating bacterial cell, which was named Synthia. And the scientific name of this synthetic bacteria was Microplasma laboratorium. In 2013, US Supreme Court had put a rule on patenting of naturally occurring genes. The US Supreme Court rules that naturally occurring genes cannot be patented. In 2013, researchers were developed functional human liver tissues in the lab. Researchers in Japan developed functional human liver tissues from reprogrammed skin cells. In 2014, synthetic yeast chromosome was created. An international team of scientists reconstructed a synthetic and fully functional yeast chromosomes. In the history of biotechnology, you can see a lot of research. Only few of them is listed here. And presently, due to the availability of wide range of tools in biotechnology, the biotechnology is used in almost all sectors of life, including agriculture, health, forensic, food, marine, etc. And presently we are facing pandemic COVID-19, and the method used for the detection of COVID-19 and its causative agent coronavirus. The technique used for this is a real-time PCR, and we are eagerly waiting for a vaccine against this coronavirus. The technique used for the detection of COVID-19 and the vaccine development for the COVID-19 is also comes under the application of biotechnology in medicine. This is a small description about the history of biotechnology. So thank you for the listening. Thank you.